Welcome to the World Congress of Pharmacology. Just a few points. If there is a fire alarm, we're not expecting it, so the exit's back there. And when we reach the end of Klaus's excellent talk, we're going to have questions, Q&A for a bit, and hidden behind the pillars here in the middle are some microphones for you to go to. Please, when you do, announce your name, say where you're from, so we know you, get to know you over the next few days. There's lots going on today, and obviously you've probably all downloaded the program onto your app. Just to draw a few things for later today to your attention, there are excellent sessions following this one. There's one on novel trial designs from Acuvia. There's several for early career researchers in medical education and statistics and training in the Middle East. And then there's also this evening an excellent couple of symposia, one of which is a symposium with new data on the beta-adrenoceptor. And the app follows sequentially, so you'll be looking at parallel sessions. The next one following this one starts at either 9:30 or 9:45. So welcome to you all, and it's my delight and pleasure to welcome Klaus Pantel here, who has very kindly agreed to talk to us about liquid biopsy establishing a new diagnostic concept in oncology and maybe a treatment concept. To introduce Klaus to you, he graduated in 1986 from Cologne University, and after a distinguished postdoctoral career in the United States, he then became chairman of the Tumour Biology Institute at the University Medical Center, Hamburg-Eppendorf. And he's really had a glittering career with many high-impact papers, and most recently, he's dedicated himself to exploring blood-based liquid biopsies in large consortia, and he's going to share with us today his insights into how we can use this in the treatment of patients and perhaps to select therapies. Klaus, you have the floor. Thank you.
Well, thank you very much for the kind introduction, for the invitation to this excellent meeting. It's really my pleasure today to talk about liquid biopsy, and I'm not frequently, or I would say I'm rarely talking in front of pharmacologists, so it's really a great pleasure, really, to talk about this new diagnostic concept to you today. These are my disclosure informations. So what is liquid biopsy? We have actually started in 2010 to call it the analysis of tumor cells circulating in the blood of cancer patients, but very soon it has become adapted also to tumor cell products released from tumor cells, in particularly tumor cell-free DNA, but also microRNAs or extracellular vesicles, and we have also included now the host cells into that definition. Why do we need it? Well, tissue biopsy is, of course, the standard way to get diagnostic information in oncology, but they are invasive, and some of the locations are difficult to access. Also, if you can access a single lesion, like a skin lesion, for example, it may not represent the metastasis in the brain or in the lung. So the blood is a pool of tumor cells and their products that are released from different sites, and it gives you more information maybe than a single biopsy. And last but not least, the most important probably advantage is that you can take single sequential blood samples over time, every few days or weeks or months, without harming the patients, but it's really difficult to take sequential tissue biopsies, so you can use that for real-time monitoring of tumor evolution and therapy response. So the vision here is to get more comprehensive and real-time information about what's going on in a cancer patient. It has been highlighted by Diana Romero a few years ago in the latest Nature Milestone edition, and I think we were very happy to see that liquid biopsy analysis has really received that recognition. Where are the tumor cells and products come from in a cancer patient? Well, in the early stages, the primary tumor is a major source of tumor cells and products released into the blood circulation. At later stages, lymph node metastasis will contribute, and at more advanced stages, distant metastasis, for example, in the bone, the liver, or the brain, will be the source of these tumor cells or tumor cell products. The blood is, of course, our main fluid that we analyze, but there are also other fluids that can be analyzed, and I will not focus on that today. But for example, the cerebrospinal fluid in patients with primary or metastatic brain tumors, or the urine in patients with urogenital tract tumors. Coming back to the blood. In the blood, we have the cellular fraction. In the cellular fraction, we can analyze circulating tumor cells and the circulating host cells. And in the plasma fraction, we can analyze everything that is released as products into the blood, in particular the circulating DNA, but also vesicles, proteins, RNAs, or even tumor-educated platelets. Circulating cell-free DNA has become one of the key analytes of liquid biopsy analysis, and we have published this review already more than 10 years ago, showing actually that the major source of these circulating cell-free DNA are apoptotic tumor cells releasing their cargo into the circulation, and of course this tumor DNA is diluted in a sea of normal DNA and can be detected by the fact that tumor DNA has certain characteristics different from normal DNA, for example, mutations, methylations, and other features. There is also another mode of release, which is secretion by extracellular vesicles, which occurs but seems to be a more minor mode compared to the release by apoptotic tumor cells. So the concentration of circulating cell-free DNA is really an analytical challenge, the same for the circulating tumor cells. So in patients with advanced metastatic cancer, you may have 1%, 5%, or even 10% tumor DNA. But in patients with non-metastatic cancer, this concentration goes below 1%, usually even below 0.1%. So we need very sensitive assay conditions. And also, if we become too sensitive, we may run into a background of mutations that people at a certain age have. All of us, if we get over a certain age, have, for example, mutations in our leukocytes that may not lead to cancer at all, but that could really, of course, impair the outcome of your analysis. So we have to be sure, if we are becoming super sensitive, that we are not detecting, let's say, a background of aging rather than the development of cancer. So the two main analytes are circulating DNA and circulating tumor cells. For the circulating tumor cells, we can enrich these cells. They're occurring at frequency 1 in a million blood cells. Then we can identify them at a single cell level using immunocytology or PCR technologies. And then we can even pick single cells, and we can do a genomic or transcriptomic analysis. And we have also developed functional models, which are cell lines that can be used, for example, for drug screening. What is very important, since these technologies are not standard technologies, we have to really perform technical validation, and we are doing that in our international consortia, and we publish the results, like here on circulating DNA, circulating microRNAs, and CTCs. And I think that's very important to harmonize these technologies to make them fit for clinical purposes. We have also performed actually or contributed to guidelines. These are the guidelines from the European Society of Medical Oncology, largest medical oncology society in Europe. And we have really put down really the different applications and how far are we with these applications from clinical practice, literally saying that in patients with advanced cancer, where you have 1% or more DNA, very higher concentration, these technologies can be used today to get information on therapeutic targets if there is no tissue biopsy available. And I think this was probably, let's say, in one sentence, the essence of a 10-page paper, but I think the paper is super important if you want to read more details. In order to actually bring discoveries or all these beautiful papers into actually clinical use, we have established a consortium, which is the European Liquid Biopsy Society, and that was initiated by support from the European Union. And we are now 75 institutions from academia and industry, and we have different working groups focusing on education dissemination, on clinical trials, on technologies, also bringing the right technology and the right clinical application together, and on regulatory issues, because, of course, if these things are introduced into clinical practice, somebody has to pay for it. So if you're interested in that consortium, you can drop us a mail. We are open for new members, and I think it's very important to close the gap between research finding and clinical practice, because there are 10,000 publications on biomarkers, but at least in oncology, not 10 new ones introduced in the clinic the last 10 years. We are also a fun group, just to say that. Of course, we need really the red chili pepper band from yesterday. I made a note for our next meeting, but we had a good general assembling in the south of France, and as you can see, everybody was happy. So what are the clinical applications? Well, these are the applications that I've shown here on that slide. The first one is early detection of cancer. You are well aware of the NHS trial with the GRAIL assay here on more than 130,000 UK citizens. So early detection of cancer is, of course, a very important application, but it requires really quite some efforts to get a reliable test. The second one is for patients that have cancer. We want to know, is this cancer cured by the initial therapy or does it come back? And I will show you data that ctDNA can be used in CTCs to get an idea whether there is an early recurrence or not. The third application is tumor evolution. We know that our therapies, of course, affect the tumor burden, so the initial primary tumor composition may not be relevant 5 years later when the patient gets a relapse, so we want to know what is changing over time due to the therapy and the natural evolution, and then adapt the therapy. And the last one is minimal residual disease, which means really in the solid tumor that after the initial surgery or radiotherapy or adjuvant therapy, is there tumor burden left over that cannot be seen by radiological imaging, and is this tumor burden relevant to the outcome of the patients? So let me focus first on early detection of cancer. We have published a couple of years ago a paper where we have looked at a single protein. It's a stress-related protein, cellular communication network factor 1, and we measured that in the plasma of patients and controls. And as you can see here very nicely, we have a higher level in the patients with cancer compared to the healthy controls. And the nice thing was also that this test already detected early stages of cancer, small tumors, not just large ones. But the AUC is, of course, 0.9, which is nice, but there's still room for improvement, so this might be a factor that can be added to a more complex biomarker panel. In order to really go deeper into early detection, we have just started a European-funded research project which is focused on the early detection of pancreatic cancer. I mean, probably everybody understands that early detection of pancreatic cancer is important. Only 5% of these cancers are detected at an early stage at T1 tumors. Most of these patients die because the cancer is detected too late. So there is a very urgent need, really, to develop more sensitive tests to detect this at an early stage where it's still curable by surgery. We are actually 17 partners from 8 countries, and we have 5 years, and the whole thing is co-coordinated by my colleague and friend Matthias Löhr from the Karolinska in Stockholm. So this is, in a nutshell, what we are doing. What we have is we have biobanks from patients that have early-stage pancreatic cancer, and we have also samples from patients that are at risk to develop cancer, so we have really 10 years or more follow-up of these patients. Then we have experts on different analytes of liquid biopsy in the blood, so we look for DNA, for ctDCs, proteins, microRNAs, and metabolites. And then we put that together with an AI logarithm to hopefully identify a classifier that can distinguish that can identify this cancer at an early stage, and also tell the patients at risk when it will be necessary to undergo surgery. We have not only laboratory researchers included here, but also health economic researchers and patient organization, because it's also very important to see what happens if you, for example, tomorrow have a test where a patient comes and says, doctor, my blood test is positive, can you please find my tumor? And I mean, you would at least know where the tumor should be located, right? I mean, at least get some idea where you should really do order some imaging, and then the imaging might be not really sensitive enough to find anything, so this patient has to come back. And I think you do not need a lot of imagination that these tests are really great, but they could also totally overwhelm our health care systems if we don't really talk and really analyze how we can introduce them in a sensible way. These are the people here in Hamburg that contribute. So just to tell you that this is a very nice translational effort between tumor biologists working on liquid biopsy, the surgeons, the oncologists, and also our healthcare researchers that look at this aspect. The second application that I think is very interesting is also early detection, but not early detection of an unknown disease, a cancer, but early detection of relapse or minimal residual disease in patients that have cancer. The advantage here is that these patients have already cancer. You know also which type of cancer, whether it's breast or prostate or lung, and you can also design assays which can be personalized to the molecular composition of this particular cancer. So you know your enemy, which I think is easier than early detection of cancer, of primary cancer. And as you can see here on that graph, our liquid biopsy methods are about 2 to 3 logs more sensitive than radiological imaging, and we have the chance here to get early signals of relapse, and maybe then we can really do a post-adjuvant interception and block the transition into full-blown metastatic disease, which in most instances is not curable anymore. So what we have done, we have looked at circulating tumor cell counts at diagnosis, and we have shown in many different tumor types here, and particularly in breast cancer, that it's associated with an unfavorable prognosis that has been also actually used in the staging, but not really widely used in clinical practice at all. But I think it's a great tool to study high-risk populations that are really improving the current tumor staging. We were also interested to see if patients with brain tumors also release cells in circulation. There's a blood-brain barrier, so it was really not clear if these cells circulate, and patients with brain tumors, like gliomas, rarely have metastasis, so it was totally unclear whether we would find anything in the blood. But we were lucky, we could find in about 20% of these patients these circulating tumor cells in the blood. They were identified by a glioma-specific stain, and then we could also show a molecular aberration specific for these tumors, so we could really prove that these were tumor cells circulating. So the blood-brain barrier is no Berlin Wall for CTCs. It has some breakages, cells can go through. But one of the questions, of course, why are distant metastases so rare? Do these patients die too quickly? Do these cells have no chance to develop into distant metastases? Or are they not really adapted to any distant organs? Are they so much adapted to the brain environment? Another question was if there is certain molecular immune composition in the brain that can help the cells even to disseminate and go into the blood, and that was a publication with our neurosurgery department again, which has shown very nicely that in patients that were CTC positive, there was, of course, a lower, I think, concentration of CD3 positive tumor-infiltrating lymphocytes and also CD8 positive lymphocytes. So the immune composition probably can also help to control the release of the cells into the blood. So the CTC research can also help, besides looking at therapeutic targets, to understand tumor biology, and we can understand the discrimination maybe of indolent versus aggressive cancers, understanding cancer dormancy. Why does it take in breast cancer 10 or more years before a relapse occurs? Is there anything happening during these 10 years? We can investigate epithelial mesenchymal transition, and we can understand mechanisms of metastasis. And we have focused here really in that context on the bone marrow as a very important organ where cells can grow out into metastasis, but it can be also a reservoir from which cells can leave to other organs, and we have a nationwide consortium focusing on that aspect. So, coming back to the early detection of relapse, what can we really do to monitor the blood to detect this relapse earlier? Let me show you a few data. In that study on breast cancer, and these were all non-metastatic breast cancer patients, we have taken the blood 2 years after adjuvant chemotherapy. The cells that we found are not derived from the primary lesion because the cells only circulate for a few hours. So these are really cells from occult micrometastatic lesions. And that hypothesis is also supported by the fact that these patients that had the CTCs had a worse outcome than the patients that had no CTCs. But you see, it's not optimal. We also miss some patients in the blue line that have obviously some CTCs that we don't see yet, but at least we see that we can pick up a signal that is clinically relevant for the outcome. The same is possible for using circulating tumor DNA. Here a group from England, Jackie Shaw and Charles Coombs, who has applied an assay for detecting circulating DNA. They have shown on the left side that if they detect the DNA, the red curves, that the patients have a worse outcome. And on the right side, they have shown how early can they detect such a signal in blood compared to the radiological imaging. And there is a lead time, there can be a lead time of more than 700 days. So it seems to be that that's really possible to detect early, and maybe now we can set up clinical studies to intercept in that phase and maybe even block full-blown metastasis. So with that in mind, we have now a second large-scale European grant, which is called Guide MRD. And here we focused really on detecting minimal residual disease by liquid biopsies that has started in May of this year. And the project co-coordinator is Klaus from Aarhus University, and the project lead from the industry side, because it's a public-private partnership, is Maria Karazarides from BMS. What are our objectives? We want to create reference standard and benchmark ctDNAs. We want to really harmonize these assays and see which kind of assays are really reliable, picking up these early signals of relapse. Then we want to do a robust clinical validation of ctDNA-based MRD in colorectal, pancreatic, and lung cancer. These are our model tumors. And we want to integrate that as a decision tool to guide treatment selection. So we have a strong collaboration here with academic and industry partners. I think we also have a very good integration of patient organization and also a stakeholder engagement. So I think it's also important to include these different key players to really get these things into clinical practice one day. Another aspect that is really super, I think, topical right now is immunotherapy. As all of you know, immunotherapy has changed the landscape of therapy, and particularly in melanoma and lung cancer patients, but also bladder cancer and other cancer patients. But of course, only a certain fraction of patient responds to immunotherapy. It's not that all patients respond. For example, 20 to 30% of the melanoma patients, even in metastatic disease, the life can be prolonged, but for 60 to 70% it cannot. So we have to find out which are the patients that respond, and also why do the patients go and develop resistance. So we are doing that by combining CTCs and ctDNA, and I'll show you some examples. And we have also shown that some of the host factors, for example, the coagulation system, can affect the development of resistance. We are doing that in a consortium together with Tatsuko Honyu from Kyoto, who has invited me to join his international network. And I'm very happy that he has contacted us and visited us in Hamburg, and that we are developing this new center of immunotherapy immunobiology. So, coming back to our circulating tumor cells, these are the data in patients with non-small cell lung cancer. We can find these cells here in around 40% of the patients, and in 78% of the patients we could detect that these cells express the PD-L1. PD-L1 is one of the major immune checkpoint inhibitors that block the recognition by the T cells and that can be targeted by antibodies that are available as therapeutic agents. What is also nice and interesting, we can also look at intra-patient heterogeneity. So it's not that patients are black or white, you know that very well. In medicine, we like to do that. We say, oh, you're positive, you're negative, boo-boo, you know, black, white. But tumor biology is unfortunately not black, white. And you can see that here. This is the same patient you see with pan-cytokeratin. That is a tumor marker, and the first cell expresses PD-L1 very nicely, the blue staining, you see that. But in line B, you see also 3 tumor cells, 1 cluster and a single cell, but they do not express this, right? So you see there is intra-tumor heterogeneity, and this intra-patient heterogeneity, because it's the blood, and this intra-patient heterogeneity can be a very good escape mechanism for tumors, right? The more different subclones I have, the easier it will be to escape some specific therapy. So that's also something which probably can be included in future thoughts. Another immunotherapy that we did in collaboration was with our colleagues in Bergen, Karl-Henning Kalland and colleagues, where we looked in patients with metastatic prostate cancer. And in Bergen, these patients got a dendritic cell-based cryoimmunotherapy. And we measured the circulating tumor cells, whether they could really predict the response. And we had 3 groups. One group had a high value of circulating tumor cells in 7.5 ml, and these patients had showed a decrease of their tumor cell contents. Then we had a group which had a moderate low amount of CTCs. Here the CTCs disappeared. And in the third group, we found no CTCs at the beginning of therapy, and they also stayed negative. And that translated also in a survival advantage, both for the overall survival and the progression-free survival, so that CTCs could predict here the responses. Then the last example is from our wonderful collaboration with the University Skin Cancer. And you can see here that was on our last meeting that we did in Hamburg on liquid biopsy. And in the background, the building in the center background is our new opera house. So if you come to Hamburg, you have to see the opera house. And you can also see there's blue sky in Hamburg. Some people do not believe that was not modified, this slide. It's original, okay. So coming back to the research, we have in that collaboration 2 cohorts. One is so-called adjuvant. These are stage 3 high-risk patients who have no distant metastasis. They get double immune checkpoint inhibition. And we have a metastatic cohort which have distant mets. They also get this inhibition. And we take blood samples in a very, very dense way, as you can see, every few weeks. And we analyze them for different liquid biopsy analytes. And I think based on the wonderful collaboration and the strong motivation of the clinical scientists, this biobank has now more than 10,000 specimens, and that is a wonderful resource for research projects. For the circulating tumor cells, we can now pick these cells. We can use melanoma marker like MCAM or NG2. We can use exclusion marker like CD45. And then we can pick these cells, we can amplify the genome, and we look for mutations that are relevant to therapy, for example, the BRAF V600E mutation. Of course, we can also do that at the ctDNA level very nicely when we take the plasma. We don't need the cells here. And here in our metastatic cohort, we have done that with the tests that look at 5 different genes: BRAF, EGFR, KRAS, NRAS, and PIK3 kinase. and we have done then a very sensitive analysis to detect really these fragments at a very, very low concentration. And when we do that, we can really show that the patients here with metastatic disease who have no ctDNA at baseline had a better outcome than the patient that had ctDNA at baseline. And it was, of course, only a pilot study, but I think promising. And what is nice, if you look at individual patients, you can see already here we have an increase already in the tumor cell DNA before the radiologists really show that there is a progressive disease, and these are the black arrows, these are the radiological imaging time points. So you get a bit earlier this information in the metastatic cohort, which may help you to change your therapy earlier. The other way around, you also can see that some patients start with a higher DNA concentration that goes then down during treatment, and also you get this information before you get the radiological information, which is shown on the right side. You see the liver metastasis that really show a very beautiful response to immune checkpoint inhibition, something you had never seen before in melanoma patients before we had the immune checkpoint inhibition therapy. Of course, you can also enlarge really the information you get. Here we have a new test that can detect up to 800 variants. It takes a primary tumor, then it designs the right analytical tools to follow up the patients. So you can do beautiful tracking over time, and it's personalized to the features of your individual cancer patient. And if you do that, and you really can also see that you have a decrease in the DNA, which is associated with clinical diagnosis of response, and that is a very complicated picture. An easier one, I think, is on the next page. On the right side, you see the Kaplan-Meier survival. So when we had a ctDNA clearance, I think we could really see all the patients were alive, and when we had no clearance, there was around a 50% death rate in this group, in this small, of course, cohort, but it's very promising. And on the left side, you see that you can detect not only 5 genes, but you can detect a whole number of genes, and it may also help you to find new druggable mutations that are relevant for these patients. Another aspect is that the tumor response alone might be not deciding whether the patients run into resistance or not. It might be also the host response, and in particularly the coagulation system interacts with the immune system in a very interesting way. And what we have done is we have looked at the von Willebrand factor, and we could see that it was a biomarker for resistance to immune therapy in metastatic melanoma that was just published a few months ago here, and which is really interesting. And you see here on the box plot that the patients that had primary resistance had higher levels of von Willebrand factor, and this also translated in different survival. So in order to bring really these assays into clinical studies, we need really clinical utility, which means we have to show that the assay can change the therapy or the management of the cancer patient, and that translates in a benefit to the patient. So one example, we've done a study in breast cancer, metastatic breast cancer, where we looked at the expression of the HER2 oncogene on the circulating tumor cells. And what we had observed is that there was a switch of the HER2 status from the primary to the metastatic time point. So in about 10%, 20% of the patients were HER2 negative in the primary tumor, but when they develop a metastasis, maybe 5, 7, or 8 years later, their circulating tumor cells derived from the metastatic lesion were HER2 positive. So we designed a randomized multi-center trial to see whether these patients that were initially negative but became positive later would profit from an anti-HER2 therapy, in this case with lapatinib. And the results, which are not published yet, are shown here. You see here the red curves are the overall survival for the patients that received the standard therapy plus the anti-HER2 therapy. The blue curve is the standard therapy, and we saw a significant difference. And I guess that is the first randomized interventional study which at least suggests some utility of CTC characterization. Finally, I would like to say, besides circulating tumor cells and DNA, there are other analytes which are super interesting. Another class of analytes that is also functional are the extracellular vesicles. These vesicles are released from the secreting cells, the tumor cell or the host cells. They are taken up by the recipient cells, and they can change the biology of the recipient cells. And these vesicles contain DNA, RNA, proteins, lipids, and we have shown many years ago with David Leiden in a paper that the composition of these vesicles can also guide organ-specific metastasis. And this was a paper together with our neuro-oncology group where we have looked for the vesicle DNA context. The vesicles may also contain DNA, and here they did a methylation profiling of the DNA that were in the captured vesicles, and that really contributed nicely to a classification of these tumors. What can be also done is a whole proteome analysis of these vesicles. This was done together with our mass spec facility in Hamburg, and we have enriched the EVs from 1.5 ml of blood plasma, and the mass spec facility could detect more than 5,000 protein in these purified preparations. And then, of course, you can do a beautiful analysis of the different functional groups that are detected, and you can compare then cancer control or cancer with and without resistance to therapy nicely and get really quite some information. So, in conclusion, I think early detection of primary cancer and MRD relapse requires very sensitive assays. The assays need to be validated and harmonized. The assays can provide clinically relevant information. Combination of different biomarkers may lead to more information and higher accuracy. Resistance to immunotherapy may depend on both the tumor and the host response, and the latter one is frequently overlooked. Or there are two groups of researchers, one focusing on the tumor, one focusing on the host, for example, the immune system, but I guess it's important to bring it together. And we need interventional studies to demonstrate the clinical benefit of liquid biopsy for the cancer patients. And we are currently discussing with the ERTC RESIST group how to introduce this measurement into the current RESIST criteria, which are based today only on radiological imaging, but they are used worldwide to assess therapy response, as most of you probably know. So at the end, I would like to say there are also many application areas of liquid biopsy beyond oncology. I have no time to go here in any of the details, but just to let you know that this field is expanding beyond oncology, and I'm very happy to see that. So, at the end, I would like to acknowledge the Liquid Biopsy Research Network at UKE that we established there in the last 20 plus years, a nice collaboration between research institutes and clinical scientists, which I think is very important for such translational projects. This is the group here. It's still the pre-COVID picture, so I think I have to make a new one. Also, I'm afraid I will look much older on this new one, so I love the old one. But I think we are still a group of 50 to 60 researchers from 15 different countries, a bit like United Nations, and I'm very happy to guide this group. And a big thank you to the funding, and particularly the EU, who has funded the ERC grants, but also the other grants that I showed today, and the German funding organizations, which help us to support our liquid biopsy research. Thank you very much for your attention.
Thank you, Klaus. That was outstanding for giving us all these insights. So we've now got some time for questions from the audience. There are microphones just between the pillars, the second and third pillar as you go down the room, so please walk to the microphone. Yes, please. Do you want to say your name and where you're from?
Paul Insel, UC San Diego. That was a very nice talk. I was just curious whether there are attempts to identify other cells that are in the tumor microenvironment, such as, let's say, cancer-associated fibroblasts, or certain of the T cells or immune cells, macrophages and so on. Is there any effort to incorporate that into the analyses that you described?
Absolutely, yes. In our pancreatic cancer grant, for example, we will do that. We will look at cancer-associated fibroblasts, because there are many fibroblasts in the pancreatic tumor tissue, you know. And we also will look at immune cells, different immune cell composition, because I definitely believe that also the host response, you know, could really reveal, for example, early development of cancer or relapse. And also it's super interesting that these host cells can actually build clusters with the circulating tumor cells. There is, for example, work from Nicola Azzoto from Zurich. He has shown that you would always think if an immune cell binds to a tumor cell, it will kill it. But he has shown if the neutrophils bind to the CTCs in the circulation, they can even help them to survive and grow out. So to understand these interactions can be also very exciting and lead to new interventions. Yeah.
Just a quick follow-up. Can one measure mRNA, just mRNAs for, let's say, certain genes that would then be possible to incorporate that may be coming from these other cells? Is that doable? Are you doing that?
Yeah, it's also doable. We have done a lot of microRNAs in the circulation, but you can also do coding RNAs. You just have to make sure after you take the blood samples, you have to stabilize your blood sample.
Sure.
So it's the old problem with the RNAs.
Exactly.
Yeah.
Okay, thank you.
Good.
Klaus, there's a question behind the pillar there. You may not be able to see the gentleman.
No?
Please, far away. Yep, good view.
Okay.
Well done.
Hi, I'm Matthew Duke from Christchurch, New Zealand. I was particularly interested in your allusion to the low-risk groups, which are a high burden on the patients and on clinics in terms of following them up, and whether, in terms of using it to define cure and potentially early discontinuation of burdensome treatment like checkpoint inhibitors.
Well, I think that we are really going to the adjuvant group, the earlier patients, you know, and we are really going now to design really interventional trials where we will discontinue or switch to another treatment based on the measurements of the circulating DNA. And these trials, of course, will tell us whether this information is clinically relevant. I think there are several of these trials now going on, also in breast cancer, in colorectal cancer, but I think we need to see the results of these interventional trials, right? There's a lot of observational trial information, and some people, I mean, draw too far-reaching conclusions from observational trials.
Thank you.
Question here.
Please.
Dr. Pantel, a very nice talk, and I'm excited to hear that you can do the immunocomposition for the biomarker for early detection of cancer, or maybe more to the chemo or IO response prediction. So you show very nicely that you could do the CD3 or CD8 tumor-infiltrate leukocyte as a biomarkers. And my question is whether we can do those CD marker in the blood leukocyte. For example, could we use more marker like PD-1, TIM3 positive on the CD8 for more precise prediction?
Oh, absolutely. I mean, what we are doing now with our new immunophenotyping is really a much more elaborate distinction of different clusters based on these markers and other markers. So we get a more detailed information. And what we are also doing is we're comparing, let's say, the leukocytes in the tissue to the leukocytes in the circulation to see, I mean, how well is it really reflected in the blood? I mean, not everything is, of course, reflected in the blood, because in the tissue, of course, you have spatial relationships, which also are very important for the biology, right? So yes, the answer is definitely we go in more detail and we compare tissue and blood and see what can be actually, what information can be deduced from the blood. Yeah.
Okay, thank you.
Okay, so Klaus, I have a couple of questions. One is, you alluded to during the talk that it's possible that the molecular signature of the tumor itself may evolve as time goes by. Does that mean you foresee us having to develop multiple assays that evolve over time to detect residual disease or recurrence? Because the drivers within the actual tumor that's shedding the DNA or the cells is evolving. So is this going to be a dynamic liquid biopsy?
Absolutely. That's a fantastic comment or question. It is actually a bit of a pain in the neck of the people. I mean, some people make the world a bit easier, too easy. They say, we do a personalized treatment, we take you as a tumor, at a primary tumor, and then we know how to follow you up. That's not true, because as you say, some minor subclones can emerge to become major subclones based on the selection pressure of the therapy. So yes, we have to actually look into that and do dynamic, I would say, profiling. Absolutely, I agree with you. So in our, for example, melanoma study, we were lucky because these patients had lymph node metastasis, no distant mets, and we could then do the lymph nodes, for the typing and designing of the follow-up tracking primers. So that was okay. But you're right. There is dynamic in it.
Yeah. And then the other question would be, do you see? So I noticed in your talk you were doing both tumor-specific, the pancreatic detection assays, and also some multi-tumor. What do you see as the future? Is it multi-tumor, or are we going to have lots of different tests?
Well, I think if you have a multi-tumor test, that's okay. But I think the multi-tumor test need to tell the doctor and the healthcare system where the tumor could be located, right?
Yeah.
So without that information, I think our healthcare system and also the poor patients will be overwhelmed, I would say, and frustrated. Yeah.
Yes, so you'll be aware that we've got a 140,000 patient study of the GRAIL assay underway, and our National Health Service is committed to 100,000 tests if that's successful. Are we losing the plot?
Well, I think you need these high numbers because the Pathfinder study has been just presented at ASCO, where they already looked at, 25,000 or something like that.
Yeah.
But then you only have 80 or 87 events, right? It's unbelievable. I mean, if you look at one time point, you take a blood sample, you have to look at really more than 100,000 to get any reliable information. And to be honest, that might not be even enough.
Yeah, that's possible.
Yeah.
So Klaus, on behalf of all of us, thanks for a wonderful talk and for opening an area that pharmacologists may not be familiar with, but will be one to watch for the future. And congratulations on a super body of work. Thank you.
Thank you very much, really.